Sometimes the passage of chromosomes from one generation to the next during meiosis doesn't go very well and can lead to incorrect numbers of chromosomes ending up in the gametes and then eventually into the zygote. So this is usually a result of something called a non-disjunction. So break this word down, right? A junction is when two things come together. So a disjunction would be when two things come apart. That's what happens during meiosis, right? The two homologous chromosomes normally come apart during anaphase of meiosis. So a non-disjunction, non meaning it doesn't happen, is when they don't come apart even though they were supposed to. In humans, there are some chromosomal disorders that we tolerate to birth. However, there are not very many. Plants can tolerate genetic changes a lot better than animals do, and so there are more chromosomal non-disjunctions that are persistent in plant species than animals. So here is meiosis 1. We can see the two pairs of homologous chromosomes separating. So in this example on the right, the two homologous chromosomes separate correctly. And on the left here, we've got a non-disjunction. And this is usually going to occur because there's no spindle fiber here connecting to this chromosome. And so when these get pulled apart in anaphase, we're going to end up with the wrong number of chromosomes in this cell. So over here, we have 3 sets of chromosomes and only 1 set of chromosomes. And over here, where it did go correctly, we have 2 and 2. This is an example of another place that non-disjunctions can occur in meiosis 2. So over here, we have anaphase 2, where again, there's no spindle fiber here, and so this is not going to pull this chromosome apart, and we're going to end up with the wrong number of chromosomes. When there is a non-disjunction in meiosis 1, all of the gametes end up with the wrong number of chromosomes. So you can see that we use the number n again. Look up here. This is our diploid cell. 2n is equal to 4. N for this cell is equal to 2. So this gamete ends up with n plus 1, which is 3 chromosomes. And these two are going to end up with n minus 1, which is 1 chromosome. If these become eggs or sperm and then eventually go on to fertilize and make a zygote, that zygote is going to have one too many or one too few numbers of chromosomes. In meiosis 2, the non-disjunction doesn't have quite as big an effect. These two end up at haploid n equals 2, which is great. This one ends up at n plus 1, and this one ends up at n minus 1. And so these two gametes are going to cause a problem if those are the ones that become the eggs or sperm. So when this happens, it's called an aneuploidy. So this is a result of a non-disjunction where either we have too many chromosomes or too few, and it's specific to a particular chromosome. So a monosomic zygote, or a monosomy, will have only one of a specific chromosome when they are supposed to have 2. A trisomic, or trisomy, is going to have 3 copies of a chromosome when they are only supposed to have 2. So where a monosomy or a trisomy is going to be a specific chromosome, so you could have a monosomy of chromosome 1 or a trisomy of chromosome 21, which would be Down syndrome. A polyploidy is where we're going to have, poly means many, more entire sets of chromosomes. So an example, right, diploid is 2n, so for humans that would be 46. Triploid is 3n, so 3 entire sets of chromosomes. And so for a human, triploid would be 69, or tetraploid, pentaploid, hexaploid. You can go all the way up to, some types of plants have 10 or 12 sets of chromosomes. So polyploidy is not tolerated in animals. Animals don't do this. An animal that was polyploid or had an entire extra set of chromosomes would be aborted. It would never actually become a fetus. A polyploidy is common in plants, so strawberries, wheat, most of the grains that you eat are actually polyploidy. They have 4 or 6 or 12 sets of chromosomes. And that comes from hybridizations between species in nature as well as human interference. Polyploids are actually more normal in appearance than an aneuploid. Remember, aneuploidy is talking about one chromosome, and then polyploid is the entire set. And so the reason that polyploids are more tolerated than aneuploidies is because you're getting equal amounts of all of the different chromosomes. So you're getting an entire set, and that keeps the proportions of the genes in alignment. Whereas aneuploidy, you have an extra of one chromosome or not enough of one chromosome, and that throws off the ratios. So let's talk about the most common human trisomy, which is trisomy 21 or Down syndrome. So you can see in this karyotype, we've got chromosome number 21 down here, and it has 1, 2, 3 of those instead of the normal 2. This is the most common and the most well-tolerated trisomy in humans. It's about 1 out of 830 births. The frequency of Down syndrome has been shown to increase with the age of the mother. So we find that there is an increased risk of Down syndrome in women who are over 35, and it actually increases dramatically when you hit 40 and then 45 again. So this 1 out of 830 is an average. If you're having a child and you're younger than 35, the incidence of Down syndrome is actually much lower than that. And as you go up kind of every 5 years, it becomes much higher. There are several common aneuploidies in the sex chromosomes that are going to happen with a non-disjunction between the X's and X's or the X's and Y's. So one common one is Klinefelter syndrome. This is usually going to be someone who's going to present male, and they have an extra X chromosome, so an XX and a Y. This does not usually cause any significant symptoms, as this is an X can be deactivated and become a bar body and not cause any major issue. Another one is the XYY, so sometimes this is called super male syndrome. They typically don't have any other symptoms. Sometimes they might be overly aggressive or have high levels of testosterone because of the 2 Y chromosomes. 3 X's is called triplo X. And this is going to happen again with 3 X chromosomes. Again, usually healthy, no physical disappearances. Sometimes they are at higher risk for learning disability, and usually one of these X's can be, again, condensed into a bar body. The only human monosomy is called monosomy X, or Turner syndrome. When we write this one, we usually just write XO to indicate that that is a missing chromosome. So this would be somebody who would present female, and this person would be sterile. They are not going to be able to reproduce and have children. Sometimes we get alterations of the chromosome structure without actually changing the overall chromosome number. So we can get chromosomal mutations, if you will, without having completely extra or too few number of chromosomes. So here's a few examples of this. The first one here is a deletion. So this is when a section of the chromosome is deleted. So here's the normal chromosome. And this example just has these genes on it. The genes are A, B, C, D, E, F, G, H, and they're supposed to go in that order. So a deletion is when a portion of the chromosome is going to get broken apart, but it never gets reinserted. And so we end up with A, B, C, E, F, G, H, and this whole chromosome is actually a little bit shorter than the original chromosome. So that's a deletion. You can get a duplication. So when a trace is actually taken out and duplicated, so it can be put in here twice. So instead of A, B, C, D, E, we get A, B, C, B, C. And so we get 2 copies of B and C. So that is duplicated. Another one is an inversion. So this is where a piece actually comes apart and then gets switched. So instead of going in the order of A, B, C, D, it actually gets switched. So the B ends up on this side, the D ends up over here, and we get A, D, C, B. Keep in mind that the deletion, the duplication, and the inversion is only concerned with one chromosome. So like this is what it's supposed to be, and then this is what it becomes. A translocation is actually going to involve 2 different chromosomes. that are non-homologous. When it's homologous chromosomes and they're supposed to swap pieces, that's crossing over, that's normal. Non-homologous chromosomes might mean that this is chromosome number 1 and this is chromosome number 7, and we're going to be switching pieces from those. So AB from chromosome 1 ends up over here on chromosome 7, and MNO from chromosome 7 ends up over here on chromosome 1. And so now those chromosomes have genes that they're not even supposed to have. So that's a translocation. So trans means across, location. They're changing location across 2 different chromosomes. There are several common disorders that are caused by some of these mutations. One of them is called cri du chat, which stands for cry of the cat. Well, this is a deletion on chromosome 5. And an individual with this is born with a very, very distinct cry that sounds like a cat meowing. And so you can actually tell if this deletion is occurring once a baby is born. They usually die young in infancy or early childhood. They have a very cat-like cry. Here's another example of one. This is a translocation between 9 and 22. And so we end up with this swap. This is called Philadelphia chromosome or Philadelphia syndrome. So that's a translocation of 9 and 22. And there are certain cancers or, for example, leukemia, that are caused by some translocations. If a translocation is going to happen on a cell cycle control gene and it gets put in the wrong place, that again can cause problems for cell cycle and can cause certain cancers. There are some genes in mammals where it matters which parent passed on the allele. So even though the trait is the same in each parent, it would matter which parent passed on the allele. And this is something called genomic imprinting. And this is usually going to be imprinting from the mother to the child. And sometimes this is usually not about what the actual DNA says, but changes to the DNA that are made after. And so there are specific genomic imprints that turn on or off certain genes, and depending on which parent passes them on, the offspring will have those genes either on or off. Even if they have the gene, they might not express it if it's been what we call silenced. There are specific imprinting genes that are critical for embryological development. So this is going to come from the mother to the offspring. Usually the mother is going to have some imprinting that is going to code for the proper method of embryological development, and we will get into this a little bit more in some of the later chapters. And then, of course, some DNA can be transferred in organelles rather than specifically from the nucleus. So we know that mitochondria and chloroplasts in plants have their own DNA. So these are called extranuclear. Extra means outside, and then nuclear is the nucleus. Or cytoplasmic, meaning they're organelles that are found in the cytoplasm. So in humans, we would look at something called mitochondrial DNA, and these are inherited maternally. So the egg is really large, and it's going to have the mitochondria inside of it. Sperm are really, really tiny. Sperm are really, really efficient. They get rid of everything except for pretty much their nucleus. So sperm don't actually bring much extranuclear DNA with them. So all of your mitochondrial DNA should come from your mother. So there's a theory that we could find the mitochondrial Eve, if you will, and trace mitochondrial DNA back through maternal inheritance to the first woman or Eve, or to follow the path of human evolution and development.